So this is just a quick summary of some of the material from the week 1 lecture where we looked at development. So in the model organism Drosophila, the fruit fly, we looked at the development of the egg through various stages through to segmentation and segment formation. And the things we discussed were the effect of these, the hierarchy of transcription factors that affect this process. We talked about maternal effect genes, which are genes that are expressed in the nurse cells surrounding the egg prior to fertilisation. And then we had a look at these other genes called segmentation genes that affect this process of the formation of segments. So these are the segments here, these individual sections within the developing larva are called segments. So we're looking at the factors that govern the formation of these segments. And the segmentation genes can be broken up into 3 groups, which are the gap genes, the pair rule genes, and the segment polarity genes. And then following this, we talked a little bit about homeotic genes and how they affect development of body parts. So, looking at this section of the previous diagram, which is the development of the egg within this follicle, here's the cell that's going to become fertilized to form the Drosophila embryo. And these are the surrounding cells, which are referred to as nurse cells. And between the nurse cells and this egg here, this cell that will become the egg, there are these cytoplasmic bridges. Now, this leads to a common cytoplasm between these 2 cells. And the maternal effect genes, is what we're talking about here, are expressed in the nurse cells, and then the product of this gene expression is passed on to the oocyte. So there are factors that affect the expression of genes within this during fertilization that are derived from these nurse cells. So effectively, the control of development occurs before fertilization because factors are transferred through these cytoplasmic bridges from the nurse cells into the oocyte. So these factors are the first step in the development of these segments and the body plan. And the development of the larva is determined by, or is broken up into these axes, and we sort of know certain factors affect the development across different axes. And I'll show you what I mean by these axes in a diagram. So development begins with a single cell which becomes polarized along these axes, and within these, so what we're really saying is that within the embryo there are regions that contain high concentrations of these factors along an axis. So you've got a head-tail axis, which is the one we'll look at, moving from the head of the larva to what will become the tail. There's also, you can break the cell up according to a 3D axis where you've got back, front, and left and right sides, but we're going to be looking at the head-to-tail axis. So if this is the larva here, this is the anterior or the head, and this is the posterior or the tail. So the head-tail axis runs in this orientation. Okay? Now, along this particular axis that we're looking at, there are certain factors that are unevenly distributed. So you've got a concentration of things, or concentrations of these transcription factors that affect the development. But before we discuss those transcription factors and those maternal effect genes, we'll just need to point out that during the development of the larva, you've got you have fertilisation occurring at this stage here. Now this fertilisation occurs within a common cytoplasm, and then within this common cytoplasm you get nuclear division for 9 divisions to give you, hundreds of copies of the genome within this common cytoplasm, and then membranes start to form around the, around the membrane, giving a syncytial blastoderm and then a cellular blastoderm. It doesn't matter what those terms are. The important thing is you've got a common cytoplasm. So if you can imagine these expression from these genomes, you've got a high concentration of a transcription factor here that'll affect expression of these genes, and you've got a different concentration of factors here along this axis that'll affect expression of a different set of genes. So it's this gradient of factors across this head-tail axis that lead to the formation of segments. So effectively, we're talking about gradients of factors that affect development. These were initially referred to as morphogens because early researchers didn't know what they were. They were just factors that affect development. And they called morphogens because to morph something is to change something. So we now know that these morphogens are transcription factors. Typically, the maternal effect morphogens is a messenger RNA that was derived from the nurse cell. So we have gradients of these morphogens established in the developing larva, and then it's the location of these factors that will determine the fate of the cells in that region of the larva. So this is a little schematic showing the developing larva. Again, here's our anterior, here's our posterior, and this is the axis that we're looking at. And the maternal effect morphogens, which are derived from those nurse cells, through that common, through those cytoplasmic bridges, what happens is that you get the messenger RNA of something called bicoid, which is located in the anterior section of the larva. And you get the messenger RNA from a gene called Nanos, which is located in the posterior. But those genes were expressed in the nurse cells, and it was the messenger RNA that passed through the cytoplasmic bridges and is now located within particular areas of the larva. So you have a concentration gradient of high bicoid here and low bicoid here, high nanos here and low nanos there. So you've got this concentration gradient of factors. Now, because these would be translated to give proteins that are transcription factors that affect development, therefore you get effects in the developing larva. So this is a little animation just to show here is located a messenger RNA for bicoid in the anterior section, and then in the posterior section, like I said earlier, you get the location of the messenger RNA for nanos. So across this axis here, you've got factors that are unevenly distributed. Now, we're looking at 2 of these maternal effect genes, which is bicoid and nanos, but there's well over 30 of these factors distributed in different gradients along different axes in the embryo. And all of these have an effect on gene expression of those. So depending on where the genome is located within this gradient, it'll experience different transcription factors. This is the same information taken from a different textbook, shown down here, Lewin textbook. And this is just showing, again, that you've got a bicoid RNA located in the anterior, and this is referring to the nanos protein in the posterior, and there's a gradient here shown in blue for the bicoid and pink for the nanos, and then these maternal effect genes affect the expression of the next set of genes that are expressed in this hierarchy of expression, which are the segmentation genes. In particular, it's the gap genes which are the first of the segmentation genes that are affected. So this just refers to an early experiment that was performed where they had a wild-type embryo and a mutant embryo. Now this was, that they did mutation studies, mutation treatments to the embryo, and they isolated a mutant that had certain characteristics. When they took a little bit of the cytoplasm from the normal embryo and they injected it at the right place, not here, but here, and at the right timing of development, they could return this mutant phenotype into a normal phenotype. So they rescue the phenotype. So that told the researchers straight away it was the location and the timing of expression of this factor that rescued the phenotype from a mutant to a normal phenotype. And this, for instance, could have been the bicoid or the Nanos morphogen. So they knew that something was localized here in the larva, and that if you replace that in this mutant, it rescues the phenotype. So that was the first evidence they had of these morphogens, and that the timing and location was important. So getting back to our story here, we've got the anterior-posterior axes. We've got the maternal effects genes that are located in different regions of the developing larva, and these messenger RNAs clearly can be translated to give the Nanos protein and the bicoid protein. So if you look at this as a graph, you can get a graph here showing the concentration of bicoid is high and it's lower in this region here, and the Nanos protein is high and it's lower in this region here. Now, following the expression of these proteins, these transcription factors then affect the gap gene, which is, in this particular gap gene we're looking at here, is called hunchback protein. So the bicoid protein is a transcription factor that activates expression of hunchback. The Nanos protein, I think, is a translation inhibitor, so it inhibits the translation of the Nanos, of the hunchback messenger RNA. So in other words, this factor leads to more gene expression, and this factor leads to less gene expression. And that gives you a different expression profile for the hunchback factor. So effectively, like I was saying, the bicoid protein activates hunchback gene expression, and the Nanos protein inhibits translation of the hunchback messenger RNA, leading to a different expression profile of another transcription factor. So if so genes in this region of the developing larva experience high hunchback and high bicoid, whereas those genomes, the genome which will be encapsulated in the cell at some point here in the larva experiences a different concentration of transcription factors. So clearly we've looked at hunchback as an example of a gap gene, but clearly there are many gap genes. So here's just listing a couple of gap genes. We don't need to know the names, but just showing that, for instance, different gap genes are expressed in different regions because of the interplay of the maternal effects genes. And this leads to smaller and smaller segregation of the larva. And then there's these regions that are different become finer and finer as you work through development, because of the interplay of now the gap genes interplay with each other to affect expression of the pair rule genes. And then the pair rule genes affect the expression of the segment polarity genes. So you've got this hierarchy here of transcription factors driven by the maternal effect genes that are transcribed in nurse cells but passed on into the developing embryo. And then the maternal effect genes affect expression of the segmentation genes, particularly the gap genes, such as hunchback. And then hunchback bicold and Nanos affect expression of the next set of genes, such as Eve. Eve is a typical example of a pair gene. So this just shows the segmentation of the embryo being broken up into finer regions because of the concentration of these different factors within this axis here, this head-tail axis. And one that's been studied particularly well is the Eve gene. So going back to here, the segmentation genes here, the Eve gene, its expression is restricted to one region of the embryo, such as one of these stripes here, and this is just some fluorescent data showing expression of different genes. So they've got some sort of antibody to measure hunchback expression, and you can see that its expression is restricted to this region here. And I also looked, I'm not going to go into this in any detail, but we looked at some of the profile of the promoters in different regions of the embryo and looked at how different transcription factors could affect the expression of these genes through the promoters. And if you look at the Eve gene here, the Eve gene is expressed because it experiences low concentration of giant and couple, high concentrations of hunchback, and because we have this arrangement of transcription factors at the promoter of the Eve gene, these different transcription factors affect its expression. So DNA that's in this region of the larva, it experiences high concentration of repressor proteins, which are the red ones. So even though there are activated proteins there, the repressors dominate and you don't get any expression. If you're a gene in this region of the larva, then you basically exposed to repressors without activators. So again, no expression. But if you happen to be a gene that's located in this region of the embryo, you experience concentration of bicoid and hunchback, which are both activators. So you get gene expression. So that's just a little example showing how these different transcription factors affect expression of genes through the promoters. We then went on just quickly to talk about homeotic genes, and I'll just, the homeotic genes were discovered because of these large-scale mutations that were seen in the developing fly. So we're not looking at just a change in eye colour or something here, we're looking at a whole body section that changes, so that these haloteres here on this region develop as an extra set of wings inappropriately. So some mutation has caused a whole change in a body part. So it's a really large change in phenotype due to a small mutational change. So people really wanted to understand during development how a small mutation could cause such a large-scale change in body plan. And the answer to this was these homeotic genes that have such a powerful effect on development. Another example of change in body plans was anatennapedia, where you have the legs in place of antennas in the head. And I think you've probably seen that photo before. So looking at these mutants in Drosophila led to the discovery of homeotic genes. So homeotic genes, by studying the homeotic genes, it became clear that there were clusters of these genes, and what was common between these genes were a DNA binding factor, which became known as a homeobox. So this is just showing an example of one of these homeodomain proteins here, and this is the DNA binding region of the homeodomain, and this region is common to a series of different proteins which all affect body plan development. And this is just showing how the DNA binding region of the homeodomain binds to the DNA. So this is an important motive domain for binding DNA. So it's clearly a transcription factor that's been well characterized. This is just looking at the binding domain, these three helix domain in homeodomain proteins, or across different species. So if you look at yeast or Drosophila, where they were initially discovered, and frog and mouse, and even human, then these regions are conserved. Okay, so it's looking at the amino acid sequence of these things. So the discovery of these HOMEOBOX genes in Drosophila led to the discovery of HOX genes in mouse and human. And HOX genes are also very important in human development. And this is just showing not only are the genes themselves conserved, but the location of the genes along the genome are conserved. So this is showing a color-coded example of the location of the gene relative to its expression. You can see that the order there of the genes in the genome relate to the location of the expressed gene in the larva. So people don't really understand why that's occurring, but clearly the location of the genes in the genome, as well as the sequence, has been conserved across species. So effectively, by studying development in Drosophila, we understand that these gradients and axis of development is really important during development, but they also led to the discovery of these homeotic genes. That mutation in these single genes can cause changes in body plan, and it's really powerful effect that these mutations have. And there's also examples of developmental abnormalities seen in human embryos, which are a result of mutations in these HOMEOBOX domain, or these HOX genes. And one of these mutations leads to embryos with one eye in the center of the forehead, rather than two eyes. And the studies of these fetuses have really, as they've identified, it's a mutation in these HOMEOBOX containing genes that causes that. So there's just a quick summary of some of the information we covered in the week 1. Okay, thank you.